Good morning. I'm going to kind of go over meiosis. It's going to be kind of just a quick overview and a review. I'll insert a few new terms here for you, but for the most part, this is kind of a review, which will kind of be nice after the craziness that you had yesterday with all those doodles. So, all right. First thing that I want to go over are some terminology words with you. The first being gene. That is the length or a section of DNA that codes for a particular trait. So we've got many, many different genes in the human genome, hundreds and thousands of genes there. So the next word that I don't believe we've talked about is called a locus. That's singular. A loci would be plural, but this is a place along the chromosome where the gene is actually located. So it's actual location. An allele is the next word, and with that one, it's going to be the place along the chromosome where your gene is located. Oh, sorry, I'm reading the wrong thing. It's the alternate variation of a gene. Sorry, I was reading the wrong spot. So the allele is the different version, I guess might be another way to say it, a different version of that gene. So hair color has different alleles. You've got blonde alleles, brown alleles, red alleles. So now something to note, we tell you up front, in biology class, hey, everything's coded for by, this one allele or this one trait. And in all actuality, it's not actually. So there's like 3 or 4 different genes that actually code for your hair color, and so you've got it's a little more complicated than what we tell you. So just to be aware with that. The next one is a homologous chromosome. This is talking about 2 chromosomes that code for the same trait, but they may or may not be different versions. It's just going to depend. You get your homologous chromosomes from your parents. So one of your homologous chromosomes comes from mom, one comes from dad. I think we've talked about those before. So we're not going to go into the actual processes of them. Mostly because we've already gone through it, so you don't need to. But we'll kind of hit it here. Just don't forget, we have to replicate everything in that S phase of interphase. And then obviously things grow, and then we actually go through this step of meiosis. So in a nutshell, those homologous pairs line up at the equator, they separate, that's meiosis 1, then you go through the second set, which would be meiosis 2, in which each chromosome then gets divided in half, so you end up with 1 chromatid in each cell. So, all right. Um, okay, so now we've got um, your four haploid cells. A couple of things to remember with those. They're going to contain one chromosome from each pair. So in a normal cell, you'd have two chromatids. Because remember, you don't get a chromosome until you duplicate it in the S stage. So you get one chromosome, so half of the information. And then you're going to be haploid. I think we've talked about recombinant DNA before. The cutting and pasting of DNA. So you cut out one gene or a couple genes in one organism's DNA, and then you insert it into another one. Maybe, maybe not. Okay. So obviously when you're inserting genes like that, you're going to get more variation. Okay. These are two things that we did not go over before. I'm not sure if you've had these two laws before or not, but you have two laws when it comes to genetics and how things get divided when going through this meiosis. The first is the law of segregation. So when we're forming gametes, when we're going through that gamete formation, which is basically a fancy way or a different way of saying when you're going through meiosis, pairs of homologous chromosomes are going to separate. It seems very obvious, but it's stated as a law. So that when we're going through meiosis, your homologous chromosomes will separate. Like I said, it seems very silly but and kind of redundant, but still important to know about. With this particular law, the reason why this particular law is important is it makes sure that each gamete has a copy of every single trait. Otherwise, if we just randomly said, oh, you're going to get 23 chromosomes, you might end up with two copies, that you could take it as, oh, you just picked 23 chromosomes, and it doesn't matter. You might end up with two copies of this gene and no copies of another one because you've got both copies from mom and dad on this one particular gene. So this just clarifies that every single trait, every single pair is going to separate, so you get a trait for everything. So you have a copy of a trait. The law of independent assortment is the second law. What this one states is that during gamete formation, traits will separate independently of one another. And I should back up here. Obviously, on your chromosome, you're going to have several genes linked together because they're on the same chromosome. So those particular genes are linked. So when you do Punnett squares, if you do them, and they're talking about linked versus not linked, that sometimes it has to do with whether or not they're located on the same chromosome. That would imply that they're linked. Because you're not splitting apart the individual chromatid. So that makes sense that if you have two, a couple of genes located on the same chromatid, they're obviously going to be linked together. However, what that is saying is that genes on different chromosomes won't be linked. So it's going to be a random chance as to what combination of traits you're going to get, or what versions of those traits you're going to get. So if hair color and eye color are located on two separate chromosomes, you might end up with mom's hair color but dad's eye color. Now, if they were both of those genes were on the same chromosome, then they'd be linked, and so you'd end up with mom's hair color and eye color. But as long it's implying that as long as you're not sharing the chromosome or the chromatid, you'll separate. Each trait's going to separate independently of one another. So hopefully that makes sense. And then just the last little thing to note, mutations, random mutations that can happen as a result of just happenstance, just by chance. Those will provide some more genetic variation within a population. Obviously, some external factors can also encourage mutations to occur, like radiation, exposure to other mutagens. You can definitely get a better chance of having a mutation enter a population, but obviously they're going to account for new traits being brought into a population, whether it's triggered by an environmental factor or not. So this one was pretty quick and easy. It's pretty short, so hopefully this makes sense. If it doesn't, please let me know. And I think I've got an assignment for you, and good luck tomorrow at Ag, and we'll pick up on Thursday with seeds. So we're getting close to fruit, which is my favorite. So, but like I said, if you have any questions, just let me know. Otherwise, have a good day.